Good morning, everyone. My name is Carla Sanchis-Segura. I come from University Jaume I from Castelló, Spain. And first of all, I would like to thank the organizers for inviting me to give this talk, but also to apologize for not being able to be physically there with you today. The availability of large datasets and the fast development and popularization of machine learning methods are very much changing the neuroimaging field, including those studies aimed to assess sex effects in the brain. In this particular field, the hope is that these novel methods will allow us to replace the description of isolated univariate sex differences by the identification of multivariate patterns of sex-related variation, then bringing this kind of studies into a new level of analysis and integration. Studies applying predictive models to study sex effects in the brain can be grouped in two main categories. Some studies use sex as a predictor of brain and behavior variation, while others follow the reverse strategy and try to predict sex from variation in brain and behavioral features. Today, I will be focusing in this second strategy. Predicting sex from brain features might be a not goal. At the end, we have much better, reliable, and even cheaper ways to know the sex of a person than looking at its brain. So, why to predict sex? Well, in this kind of studies, the true goal is not the obtained prediction. The interest and relevance of this kind of studies resides more in finding out to which extent the brains of females and males can be accurately classified. This is so because in this case, the aim is to indirectly assess the degree of statistical distinctiveness or separateness of the brains of females and males following a backwards deductive strategy, which rationale goes as follows. If we can accurately classify the brains of females and males, that must be because those brains are distinguishable, and that must be because there are many or large differences between them. However, as I will try to show today, the use of classification metrics to measure sex differences and similarities is associated with major shortcomings. Fortunately, as I will try to show today also, we have better alternative to quantify the degree till which the brains of females and males are similar or distinct. Binary classification metrics cannot properly measure sex differences. This is so because the calculation of this kind of metrics requires the dichotomization of the continuous output provided by predictive models. Dichotomization is known to result in arbitrariness and major information loss. In fact, it transforms what it should be a quantitative measurement process in a forced choice between two mutually exclusive alternatives. Specifically, when a continuous variable is dichotomized, the cases that are close to the cutoff but on opposite sides of this cutoff value are arbitrarily treated as if they were totally different, when they can be very similar. Simultaneously, the cases that are located on each side of the threshold are arbitrarily treated as if they were totally identical, when they are not. As a result, what happens is that we lose all quantitative information, and this quantitative information is replaced just by category labels. So the use of classification metrics in sex prediction studies often leads to unwarranted conclusions. So the high classification accuracy observed in this kind of studies, usually ranging between 80–90%, have been often interpreted as indicative of the existence of large differences between the brains of males and females, or even as confirming the existence of two types of brains, male brains and female brains. However, while this might be the case, we cannot really know from the results of classification. This is so because, as already mentioned, classification requires dichotomization and categorization, which replaces all quantitative information by nominal labels, so the number and the size of the possible differences between these two groups cannot be really estimated. In fact, the same classification accuracy can be obtained in very different scenarios. So, although we want to know how much different the brains of females and males are, the answer we get doesn't answer this question. However, we can erroneously think that it does. On the other hand, finding two types of brains is not a result of classification. It's a precondition that we impose through dichotomization. It might be thought that this is justified because at the end, males and females are two separate categories. However, we should not forget that once we put males and females in any metric space, they are no longer categories, but usually they act as two distributions. So, what can we do? Well, since all the shortcomings we mentioned stem from dichotomization, perhaps they might be solved just by avoiding it. We can follow the advice of Lipa and Connelly in the 90s, who suggested to compute diagnostic probabilities of class membership and then use them to measure individual differences. We also suggest coupling this strategy with robust statistics and informative graphical methods that exploit all the informational richness contained in those distributions and then allow us to describe males and females, to compare their distributions, as well as estimate the magnitude of their similarities and differences. To illustrate this approach, today I would like to share the ongoing results we have in a project in which we have taken a large sample of right-handed participants of the HCP dataset and then obtained their estimates of great matter volume in 18 brain regions that conform the so-called language core network, which are brain areas that when lesion lead to aphasia. We obtain those estimates, normalize them, and then use them as predictors in a logistic regression model so we could calculate these classification probabilities. Before setting our predictive model, we assessed the univariate sex differences in all brain areas that were used as predictors. Because we observed that in all brain areas there were moderate to large differences favoring males, we set male as the reference category in our predictive model. In this way, large classification probability scores were aligned with larger gray matter volumes. I must say that we used a logistic regression model because this kind of model competes well with more sophisticated classification algorithms, but it's much more interpretable. In fact, logistic regression allows obtaining nomograms like this one. In a nomogram, all predictors are depicted as independent scales, which length is proportional to the relative importance. Thus, by plotting the medians of females and males in each predictor, we could illustrate where and by how much these two groups differ, and also how those differences add up, and finally how they get nonlinearly projected into a single dimension given by their classification probabilities. Classification probabilities provide a unidimensional metric space that combines the information contained in all predictors. Therefore, by assessing how females and males differentially occupy this empirically created metric space, they can be described and compared at the multivariate level. For example, in this case, we can easily observe that females and males do not form two mutually exclusive categories, but two very skewed distributions. In this case, most of the females group near the lower bound of the P-class continuum, whereas most of the males accumulate near its upper bound. However, these distributions are also very long-tailed, and some individuals spread far beyond their respective sex clusters. Consequently, the males and females distributions overlap, and by quantifying this overlap, we can estimate the overall degree of similarity between females and males in the 18 brain areas used as predictors. We can also quantify the probability of superiority of either sex, which provides an overall estimate of the multivariate sex differences between males and females. More detailed information about these distributions can be obtained by dividing them in meaningful sections. In this case, we opt for dividing each distribution in its tens by calculating the values of their deciles. When the goal is to compare females and males, empirical cumulative distributions are very useful. In this kind of graphs, comparisons can be performed in at least three different ways. First, we can choose a cutoff value and then check the proportion of cases in each group with p-class scores that are higher or lower than the selected cutoff. If we choose 0.5 as our cutoff value and then sum the proportion of males and females above and below this value, we obtain the percent of correctly classified cases. In this study, 81.8%. However, the advantage is that we can repeat this comparison selecting any other possible cutoff value, or we can even include a gray classification zone similar to that used in equivalence testing procedures. A second way in which we can compare males and females in these graphs is by assessing how many subjects in one group have p-class scores that are equal or higher than a given proportion of cases in the other group. For example, in this way, we can calculate Cohen's U3, which estimates the percent of individuals of a group that have larger scores than the median of the other group. However, once again, we could have chosen any other possible criterion relevant to our hypothesis or directly stemming from the data. Finally, we can use these graphs to compare location measures such as the medians, with the advantage that because p-class scores are naturally bounded between 0 and 1, the differences we obtain are standardized, and therefore they are easily compared between studies. However, it should be noted that difference based on medians or in any other single value may or may not properly summarize the difference between females and males. Therefore, we decided to compare all 9 decile values, then avoiding to reduce the study of sex differences and similarities to simple comparisons between the typical male and the typical female, which far too often are used to make unwarranted generic statements about all females and all males. What we observed was that differences in the central locations were significantly larger than those observed in distal locations, then confirming that not all males are equally different from all females. Yet, when the aim is to estimate sex-related differences and not so much to compare females and males, the distribution of all the pairwise differences can be calculated, and this distribution can be directly analyzed using the same 3 approaches just described. In the case of the present study, what we observed was that differences were asymmetrically distributed, with differences favoring males being more frequent and also, in the majority of cases, larger. More specifically, a difference favoring males should be expected in 89% of the cases, and the size of the expected typical difference was estimated in around 0.5, that is, around 50% of the possible maximum. By now, I hope to have convincingly shown that when the output of predictive models is not dichotomized, information about multiple brain features can be condensed in a single continuum that allows describing and comparing females and males on an standardized 0 to 1 scale with a variety of meaningful effect sizes. I also hope to have shown that this analytical strategy is highly flexible, and also that it has several advantages over other analytical strategies restricted to the calculation of classification metrics. However, before finishing, I would like to mention one more final advantage of using p-class scores. More specifically, I would like to highlight that while classical classification metrics are self-confined measures, p-class scores allow evaluating the relationship between the composite of the brain features that we are evaluating and other variables, and this allows to identify possible moderators, mediators, or confounds. Thus, for example, in this case, we observe that p-class scores are directly related to total intracranial volumes, then suggesting that the sex differences just described are largely influenced by the gross morphological differences that exist between the bodies of males and females. This finding prompted us to repeat our analysis after adjusting our gray matter volume estimates for tip variation. Today, we don't have the time to go into the details of those results. However, I hope that a quick glimpse into their graphical representation will suffice to show that after statistically controlling tip-related variation, the multivariate sex difference observed in the core language network reverse their direction and very much reduce their size. Thus, in this case, differences favoring females are expectable in 68% of the cases, and the size of the typical sex difference will be lower than 10% of the maximum possible. Yet, the result I would like to highlight is that, regardless the fact that multivariate sex differences obtained from raw or TIFF-adjusted features were very much different in direction and size, in both cases the obtained nomograms were quite similar, and then they pointed out some brain areas as being the most affected by sex. And also, in both cases, the structure of the obtained nomograms was highly correlated to the size of the univariate sex differences in the brain areas considered, then confirming that difference obtained from P-class scores provide a valid summary of those univariate sex differences. And with this last conclusion, I would like to thank your attention.